about designing DNA binding proteins today. This is work from the Baker lab and done in strong collaboration with two other people, um, Anisha Sehgal and Yulia Politanska, and also Paul Kim and many other people there. So before I start, I want to kind of talk about why DNA binding protein design is harder than things like protein binder design. It's because DNA is a charged molecule, um, so we have to displace water, and it also looks globally, generally, double helical. Um, so the variation between different sequences, or the global structure of different sequences, is very tiny. And however, this is an important question we want to pursue because it has a lot of effect in biology. Basically, the deepest part of our biology is gene expression, um, quality control of a genome, and editing of a genome. All of these things are done with DNA binding proteins. And there are existing systems like Cas, zinc fingers, which help engineer some of these things, but they have their own limitations. I won't go too deep into those things, but um, our dream is to be able to design DNA binding proteins which are totally de novo, have their own topology so we can take advantage of those things. And this is not the first time we in the Baker lab tried something like this. So there was a paper you might have seen, um, which came out in 2025 called Computational Design of Sequence-Specific DNA Binding Proteins by Cameron Glasgow, which was using pre-deep learning era techniques and sampling native scaffolds predicted with AlphaFold 2, basically, which means these are small variations of natural proteins and not totally de novo. And also, the biggest issue we had is that the design process was not really uh, taking care of specificity. So if you want to find a specific binder, you have to screen 100,000 plus designs to get one. And even in terms of getting just binders, it was not that easy. Um, so, so what changed since then? So since then, the protein design has been revolutionized by deep learning. So the general framework of protein design is like you start with a generated backbone of a protein with something like arc diffusion. And then you do sequence design onto it, sample multiple sequence with MPNN, and then you use some kind of oracle, which are structure prediction methods generally, to tell you if this sequence that you generated falls into the same backbone that you um, started with or designed for, because the backbone encodes your function generally, like binding, catalysis, things like that. So um, for DNA binding, this is how it looks like because it is not just predicting the binder, you have to predict the binder to align to the DNA at the right place. So if you design and fold some things with AF3, at 1 angstrom DNA aligned RMSD of the protein, this is a very good fit. So this would give you a lot of confidence, while this means AF3 did not think that it would dock. AF3 is not a perfect oracle, by no means we claim that, but the signal that it has, we want to enrich that to get binders in the wet lab, basically. And um, around the same time, also RF diffusion 2 came out, which somewhat solved enzyme design. And what RF diffusion 2 does is that you give it a very carefully curated active site through quantum calculations and stuff like that, and then it generates a backbone around it. So people in the Baker lab had this idea that maybe we can take this scheme and generate DNA binding proteins. So this is the second era of the attempt of getting DNA binding proteins in a specific proteins in a tractable manner. So what the idea was that, like enzyme, you can see these bidentate interactions, which are supposed to be specific to specific bases. Like only arginine can do this interaction with G, and only say glutamine can then do this with A. And then, like enzyme design with RBD2, you can scaffold those into a backbone sequence design AF3 and test them in the wet lab. However, so that's what basically happening. You here is a broader picture of the same process. You take these tip atoms, fold the helix. They're intermediate state, but in the end up through folding with AF3 and other kinds of sampling, you end up with a binder you want to test. So there was a lot of binders that we tested with this approach, like 26,000, and ended up with only 4, which basically meant that there was not a great improvement compared to the pre-deep learning era using the same approach that solved enzyme design. So around this time, so what now? I gave this group meeting in the Baker lab where we're talking about imagining the next generation of design DNA binders, which will actually be possible. How can we make it actually possible to design DNA binders, specific DNA binders in a small scale screening scenario? And the key observation, in my opinion, is that recognizing that specificity conferring protein DNA interactions are diverse and complex. So what we looked at in the RF diffusion 2 era is this thing like pseudo pairing or bidentate interactions, which is an important mode of DNA-based recognition, but there is a lot of more things going on, generally in natural transcription factors, like recognizing the slight variation of the minor group width of a AT-rich sequence, where because these backbones will come closer, there will be a highly electronegative environment, and arginine will recognize that variation, or stacking, or net specificity conferred through hydrogen bond networks like secondary supports, which lets these things recognize the shape of a backbone or things like that. And so this previous pipeline had this issue where we are seeding these bidentate interactions consecutively or quite a lot, but what that creates an funny artificial restraint on this backbone, which kind of pushes it away from where the how a naturally gripping DNA binding would have happened. So it sacrifices other kinds of specificity and affinity conferring interactions to do this thing too much. So when we realized that, we also realized that um, that we cannot make biased assumptions about how we want to recognize the DNA because it is complex. It has so many acceptors and donors. It is a flexible molecule. So we need to reduce those kind of heuristical intervention, but also we need a stronger diffusion model because we realized that this previous generation of models could not do um, well enough scaffolding of DNA binders that would give us a high enough pass rate to enrich for such kind of complicated interactions. So, so around that time, David Baker clubbed me with these two gentlemen, Jasper Butcher and Rohit Krishna, and other people in the lab who are trying to create a new style, new diffusion model. You may not have, may or may not have seen it, but it's we call RF diffusion 3. So a little bit about RF diffusion 3. We start with the architecture of AF3, taking inspiration from it. But what AF3 does, or AlphaFold 3 does, it is a structure prediction method. So it takes a lot of a rich input, MSA, template, conformer generation, and of course the sequence of the thing that you want to predict. And then it, munges on all of that in this huge, what you call like the trunk. And then it has a diffusion module, which is conditioned by the information from the trunk to generate a structure. But we realized that you could do the same thing for design, but it doesn't need to have that much of a rich input for the trunk because in design, you are not telling what the sequence should be. Everything has to have kind of the same identity of residue, and then it should diverge into different design residues. So in RFD3, this trunk is very small, while this diffusion module carries the load of designing the protein, which makes it really fast compared to previous iterations of these methods. And just to more pictorially show what happens is that you start with 14 atoms, 4 backbone atoms, C and C alpha O, and then the 10 rest of the virtual atoms will, through the diffusion process, choose to be a particular residue, and and you would put the rest of the unused atoms somewhere here. And same, so it can become a phenylalanine, or it can also become an alanine. Now, it is not necessary that RFD3 generates sequences which are as good as MPNNs, but what is important is that it generates sequences compared to previous diffusion models, which means in context of the DNA, it knows how far to put different parts of the backbone. So it should generate better backbones compared to before, just because it has a sense of how the side chains would somewhat look like. So this is how roughly this is somewhat of an artistic rendition of how that process plays out. But as you can see, RFD3 has this sense of seeing the DNA and the protein together as in an all-atom manner. So it will, when you sequence design on top of it, you will get a better chance of getting sequences which are more foldable by AF3. There was a complicated process of development because its RT3 was not just about DNA binder. There's a lot of people's work went into it, so we wouldn't go into that discussion of that a lot, but I suggest you look into this preprint we have on bioRxiv. Um, but shortly after we got RBD3 to work, I did a small scale design campaign on some sequences where I designed this kind of similar sized proteins that we would be designing with the previous versions of our design strategies. And we found that when we tested only 5 designs targeted towards this random sequence, we found one binder. So this was not a super specific binder. As you can see that it has activity in off targets, while the on target is somewhat on the higher side, but it is not super specific. Neither it was a super strong one, but now we knew that we can get binders by testing only a handful. So now we want to go from there into a design, into a design process. How can we take this diffusion model and a clever design process to, push it to the scenario where we are getting strong binders and specific binders. But this was the encouragement that we needed at the moment. So further computational encouragement that came out shortly after was that we not only we can design small binders like that, we can also go to longer length. Longer length binders for DNA binder means it's a little bit more complicated because it has to wrap around DNA and has to contact more bases. But this also had the advantage of conferring more specificity because it can directly contact more larger regions of the DNA, right? So that means it has also has a higher chance of finding a specific location of the genome. So comparing and the smaller binders from RB2 versus this longer version RB3, RB3 still outperforms pass in terms of A3 pass rates quite a large margin. And larger binders are more difficult. So with that all that juice in terms of getting passing distance for A3, now we can do more in terms of filtering for interactions that would make it more native-like instead of more artificial, as we were discussing before. So we did a lot of filtering on hydrogen bonds and hydrogen bond supporting networks, hydrogen bond backbones and major groups, etc. And we ended up with a set of designs that we are looking to order. And what is encouraging is that compared to previous um, versions, now we have for RFD3, it is very competitive to native. So these plots are showing per nucleotide interactions of different kinds. So per nucleotide, how many total hydrogen bonds, per nucleotide, how many phosphate backbone hydrogen, etc. And so at this point, we have novel scaffolds that do not look like nature by a large extent, but they interact like nature. And this is what we ended up testing in the lab. So these are some examples of the things that RFD3 based design could generate. And also, not only these major group binders, there are some sequences which are unusual, have an unusual shape, like this CGTATA, CG. This kind of sequence has high bendability. So you can also design minor group binders for these things, which could be specific. And these are some examples of RFD3 generated minor group binding scaffolds and their overlap with corresponding AF3 prediction. So before going into wet lab research, we also you might have seen this preprint came out called Bolsgen, which also can generate DNA binding proteins using a very similar scheme kind of architectural schema that RB3 uses or AF3 uses. But and we saw that RB3, does comparable or better with this competing method, which came out after RB3, which was nice to see. And then now it's time to actually test these things in the lab. So our targets are 10 to 12 base pairs, and we had initially a set of 6 core targets, which we kind of used for our development of pipeline and the initial rounds of testing. And these sequences are chosen such that they don't have, more than 4 more k-mer overlap among each other. So it is a slightly easier to distinguish between them in theory, but it would give a strong signal in terms of whether we can generate specific binder or not. And then we did a stronger test with more sequence, which were all folded against these guys and also these guys. So total of six among 16. So now that in that scenario, you have to find a binder where it does not bind to 15 other sequences. Um, and these were not used in the development process. So it's like a very clean test. So this is how the design pipeline look like. So you start with a DNA, you diffuse a protein around it with RFD3, and then this step is crucial where you are folding it, sequence design it, and then filter the bad ones out, and then folding it again, sequence design it. Why? Because DNA is a flexible molecule, and the initial DNA is not necessarily moved or conformationally packed well with the protein. While once you do the folding one time with A3, it looks more natural, and then you sequence design on top of it, and then what you end up getting is something that is more well packed. As you can see the DNA backbones here has moved in the final design, for example. And so that is where you can generate good binders, and you can actually take things directly out of this pool here before going to into this off target folding with AF3 and test. But what this off-target folding with AF3 we found to be very helpful for making it even more better in terms of screening less design. So you can test from here. You can also test after doing this off-target filtering by maximizing AF3 confidence. So basically folding its off-target DNAs and take the ones where the AF3 confidence of the on-target is the highest. And so this is kind of how the assay looks like in our initial experiments. So the x-axis is protein expression, y-axis is DNA binding. So that means anything that looks diagonal means that there was good protein expression and the protein bound to DNA. So you see signal at the same time. So this is a strong binder, while this dark gray one did not bind as well. And you can turn that into a heat map, so it will look like this, where on target is say it's well bound, off targets are not that well bound. And here is the results on the first set of 6 core targets, where we found a total of 42 specific binders, and a lot of them for specific targets came from just screening 96 designs. So we went from screening 100,000 designs to just be able to screen 96 designs, which has implications in the wet lab and how easy it is to how feasible it is to do for any lab compared to just big labs like the Baker lab. And on the test targets, 9 test target we tried, we found 4 targets for which found 1, 2, 3, 4, like about 14 binders, which were specific against all 16 of these targets that they were off-target folded against. So this was this validated that we do not have a bias in our 6 core target or initial experiment. This is like a strong test that we uh, um, in the wet lab it showed that we can now really design things by testing all these 96 designs. So yeah, this is basically kind of the summary of that. While it's basically showing that this design campaign, the average hit rate of specific designs is like 3%, while previously we had 0.01%. And in terms of affinity, we also found that all of these designs, um, range an affinity of very low nanomolar, like 3 nanomolar to 30 nanomolar. And and These are the same designs which also saw in the lab to be specific among these 16 sequences or like 6 sequences. So this kind of so by aiming to build which binders which interact like natural binders but look de novo, we were able to hit both affinity and specificity at the same time. But and also looking back into this, complicated biophysical interactions, we saw that compared to the old RFD2 orders, which would be in this blue, things we ordered, I already showed you, were similar to nature, but things that succeeded were in the same distribution. That means we did a good job of filtering on those interactions to be in the right distribution where success would come. But so far we discussed, specificity against specific sequences, like, 16 off targets or something like that. But now we also characterize more fine-grained specificity. So what you are seeing here is that for these different designs, we are showing what happens if you mutate this A to say C, G, or T. And green means that the binding was weaker than the wild type or the intended sequence. So anywhere you see green means that that position was specific to the intended base that we wanted it to be specific for, while orange means that it was off target or slightly higher. So this design here is really good, which has among 12 times 4, like 48 variants, it has like 1, 2, 3, 4, only 4 off targets, which are slightly stronger. While this design has 4 times like 7, 28, this block to be pretty specific, while this part is not that specific. And you can actually correlate that structurally with, in purple, these are F3 PLDDTs of the contacting resid. So you can see a little bit of lighter PLDT here compared to the darker part here. And that basically shows you this loop here, which is probably flexible and can move around a little bit. And that creates the non-specificity in this part. So that kind of gives us some indication of, what to look for later on in next to next round of designs and incremental improvements of this process. And you can see the same thing here, lack of contacts with lack of specificity, while these regions are very specific. Um, this particular one, I really like it. This is my one of my favorite ones where it is like really green everywhere. And actually, what happens is that, this part here, you can actually see from here that there is not a lot of contact, but it is still shows specificities to some extent to the intended sequence. And if you zoom in on that part, it's very interesting because there is no direct base contact. Well, there's a little bit of direct base contact, but a lot of it comes from the protein doing 5 consecutive in recognition of these backbone phosphates. So this is the AT-rich sequence. So the protein has to bend the DNA in an intricate manner to be able to do that. And that is possible because of the flexibility of the AT-rich core, which probably indirectly confers specificity um, towards this bound shape and this DNA sequence. We also found interesting binders where for the same target, we found complementary specificity profiles. For example, this one here to this human NF-kappa B site, it recognizes these 2 GC-rich flanks to specifically, while the middle part is not specific, while this one here recognizes the center part super specifically, which is a AT-rich part. So it is showing that, we are not just able to recognize the GC-rich regions, but depending on different designs, we can actually do all kinds of specific recognition of the same sequence in different recognition modes. Um, and so now some of these off-target effects are also validated by, both on-target and off-target effects are validated by these tools called NAMPN and DPBS. I built DPBS, and Andrew Kubani built NAMPN from the Baker Lab. And these are tools that predict, sorry?
Raktim, it's me here, Lukman here. About there's a slide, the previous slide. I think it's about sequencing specific bound
Yeah,
shape recognition, I think.
this one,
No, the next. In fact.
this one.
Eh, Yes,
Yeah.
mainly.
Yeah,
I was wondering, like, in terms of like specificity,
yeah.
uh, how much of it like can be attributed to
The
like sort of
conform the
like
shape readout.
no like direct base recognition,
Yeah,
like versus sequence in terms of
yeah.
looking at the sequence of the DNA shape recognition, like how
Yes.
much of it depends of specificity comes from the direct
Yeah.
base
So,
recognition.
the good question. In general, there's a huge literature on these things, and depending on your target sequence, it can vary. Some sequences are more amenable to sequence or chemical recognition. Some sequences are more amenable to shape recognition. This particular one, you see, so this left interface in the middle here is chemical recognition, where these two AT pairs are being bidentate recognized by bidentate interactions, and you can actually see that right here, these two pairs, while this region on the left, which is not that strongly green but somewhat green, is this part here. So in my mind, you can call it like 30%, 70%, but in general, it varies depending on what sequence you're talking about. Does it sound good?
Um, yeah, sounds good. I was biased because somewhere
Yeah.
you talked about, I think, guanine.
Yeah.
I don't know that the question links properly to that, but you said like guanine sort of tends to give some sort of an edge amongst the nucleotide base pairs. Like,
Yeah.
uh,
Yeah.
yes, so I thought the two are connected, but if not, uh, that's fine.
Sorry, say that again. I did not catch that.
Like in the mid of the presentation on, I think after
Yeah.
like the introduction,
Yeah.
you were representing how the I think
Right.
the binding, and you talked about guanine, and I
Here.
think the yes,
Yes.
so
Yes. Go on in.
I thought the two are connected.
Yes. Yeah. So guanine, this is a chemical recognition, but the same residue, like arginine, can also do shape recognition in the minor group. So they are
Okay,
connected. Yes. Yeah. So this thing here, we have multiple. I mean, I didn't want to discuss the exact interfaces of all of them, but here I can show you. For, so let's see. Um, yeah. Okay. So here is an example of arginine recognizing a guanine, which is position um, this G right here, position 3. And similarly, this is a similar thing but with glutamine with adenine, which is for T7, which is this position. It's T, but it's A on the other strand basically, and it recognizes the A on the other strand, so it's T.
sure.
Yeah. So yeah,
Okay.
so all of this, and yeah, there's also this tyrosine here, which interacts with this arginine, which will stop it from rotate away and do something else, for example, which you would come under H1 support.
I see.
Yeah. Yeah, I think the manuscript has a better discussion of individual interactions. I definitely, if you find that interesting, I would definitely recommend you check those out. But these are, yeah, in general, shape and chemical readout, I would say like 30 to 70% distribution. Anyway,
Thank
oh,
you.
so yeah, so these are methods which can predict, given a design model, with what is the specificity profile. So you may or may not have seen these logos, but these are basically showing that this position prefers T strongly, and that agrees with what we found in the experiment. While both of these models predict that this position will also prefer C, and we actually saw that from the experiment too. So um, similarly, both of these models predict this part should be somewhat non-specific, which agrees with it. But there are also disagreements between these models, and that's a whole other can of worm. But it's interesting to see that independent computational models can validate some of the specificity profiles that we see. Interestingly, this one really likes G everywhere, but these two, the models cannot predict that. So continuing on, so this discussion was about specificity, single nucleotide variant specificity. But you can have an even stronger test of specificity where you pit your binders against a random library, which is called HT-SELEX experiment. So you start with a random library of DNA, you put your protein, and then you enrich for the things that bound, and you repeat that. So you take out things that are like, strongly, really strongly binding over multiple rounds of binding assay from the random pool. And for natural TFs, you come by taking the final sequences, you can generate these kind of logos where you can see this one is like really specific to CAC-GTG, while this one is somewhat specific to TG-TGJT. And most natural TF actually look like that. And so we wanted to see how our binders look like. And so we ran HT-SELEX for with these collaborators from Juicy Taipale's lab. And we found some binders which did pretty interesting things. So for example, here, this one, targeting this sequence, which has this repeat of GAGGA. And we found that the SELEX PWM closely corresponds with this fiber motif that repeats. And so it just tells that, so this design well actually is binding to the second GAGGA, but it could bind to the first GAGGA also. So that is a cool one. It has also a reasonably good amount of information content above one. This one was one of the strongest signals that came out of SELEX, and this is a binder designed towards the CAG repeat sequence. You can say four CAGs, but it also has a GCA repeat, so GCA three times, and it looks like it recognizes the GCA repeat very strongly. And actually, on the other strand of GCA, you have TGC, right? That's the reverse complementary. And which means in the double helix frame, it looks like GCA on this strand and also GCA on this strand. And so GCA here and GCA here, and which indicates that this binder is probably binding in like multimers over these repeats, which is very helpful, potentially helpful therapeutically because these repeat sequences of CAG are related to Huntington disease, and if you can bind large areas of those repeats, you might be able to block transcription of the Huntington's diseased version of the protein. So that's something people would be interested in testing downstream functionally. Here's another PWM that I find interesting. This one is not a very strong PWM, but the top motif agrees very strongly with this AT-rich core of this target. And in fact, previous methods has 0% pass rates towards this target. This because, the AT-rich DNA conformation is generally not standard, and it was very hard to design against that. But it looks like this binder that we got for against it had uh, close overlap towards its specificity profile had a close overlap towards the intended target. Although read count, I would say the read count of this PW was pretty low. We also tested some of those interesting minor group binders, and we found that from testing like 46, we found two binders which were able to bind this AT-rich sequence from the other side. So if you cannot tell the difference, it's the same target, but it's binding on this major group side, this broad side, while this is the standard binder, while these minor group binders are bending the DNA. So instead of this side, it is binding on the other side. And these are some different topologies that we ended up testing, and these are the two that we found bound. Interestingly, both of these two were pretty close to the natural TF. So the natural solution that nature found is pretty close to the most optimum for this kind of bent AT-rich sequence recognition. Um, so summary-wise, RFD3 based design campaign with cycling and in silico filtering. This is what we present in terms of what people can use. It is computationally much lighter than any of our previous approaches, and it shows 100x better wet lab success compared to what we could present before. And the designs that we could get out of testing, 96 designs only had strong affinity and strong specificity and varying degrees of broad select characterized specificity. So future directions are, putting them in the genome. So in the in vitro versus in vivo, it is very different. So we have people working on now putting these binders in the genome and seeing how you can, whether you can, what is the genomic binding landscape and how you can, activate gene transcription or repress transcription with these binders. And also, can we, modularize them now? Now, can we link them together to make, even longer binders, which would recognize even longer stretch of genomic DNA? Yeah, so mostly that's about generative design of DNA binding proteins. I want to add a little bit of extra for you guys where about nucleic acid co-design with RFD3. So you might remember I showed you this slide a little bit earlier where we can do all atom design of protein with RFD3, but I also implemented extended version of RFD3, which can do all atom design of nucleic acids also. So basically the same concept, you have virtual atoms that did and backbone atoms. So the backbone atoms are shared, four for protein, however many for RNA, and however many one less for DNA. And then you have these virtual atoms which will design the side chain and put the rest in C1 prime. So with that, the RFD3 framework is full biopolymer design, and it can create things like this, where I tell it to design me a one protein chain, one DNA chain, and one RNA chain, and it created this protein nicely hugged with a long RNA chain with hybrid with the DNA. And well, this is the same slide. And we found that you might have seen this old RFD2 or the all-atom based method on bioRxiv. It will be published soon, which does something similar, but not all atom. And we see that the structures that are generated by RFD3 based all atom design of nucleic acids have better foldability compared to the old method. And these are some examples of intricate designs that were made with specific secondary structure constraints that had very good agreement with A3 prediction. Another thing we saw, because we are doing co-design, we're generating the sequence also. So for RFD poly, it found this interesting kind of niche solution where it would put CG pairs in always paired region and A in always unpaired on all unpaired regions, which is an interesting solution, but it's not necessarily always realistic or you what you would expect. While RFD3 solutions, you can see there is a mixed bag of, all kinds of base pairs, looks more natural, basically looks more like the PDB. And also you can, in theory, do very complicated enzyme active site scaffolding now because you can generate both the RNA. So this is basically a tRNA substrate of an enzyme which has both a protein component and an RNA component. So this is kind of the active site where you have these many RNA residues and some protein residues. And you can actually redesign or, if you design this active site, then you can totally de novo design enzymes which can be simplified version of natural enzyme. So here is an example of a diffusion output which matches the motif up to 0.36 angstrom, basically. These are not folded, just a proof of concept, but we're opening the door up to doing more complicated enzyme designs involving protein and nucleic acids. You can also do small molecule binding aptamer design, which is something that people asked us a lot about when the RFD poly paper came out, because that was not a functionality offered by that model, but I'm happy to say that RFD3 can do it. And here I'm just showing binders generated for AMP and FMN using RFD3. So this is the target molecule, and then the design is in blue, and then in gray you are seeing the AF3 overlap. This is a very difficult challenge because AF3 is not as good in predicting RNA structures as it is for protein structures for many reasons, and docking the molecule right on top of is even harder. So, but the fact that we can do co-design of that with success rates of, like 1% for less than 4 angstrom RNA aligned ligand RMST um, gives a strong in silico signal that these things would be for wet lab viable super soon. Yeah, so the code for the nucleic acid functional RFD3 is available in Rosetta Commons Foundry. And yeah, with that, I want to show this acknowledgement slide. Anisha, Yulia, Paul, Najim, Andrew K. These other people are our DNA binder generation group doing work at different aspects of it and follow-up projects of it. Tushar from Thai Paralab did all the selects. Jasper Butcher, Rohit Krishna, and other people worked with me to build RFD3 and yeah, and the IPD admin, and of course David Baker. We had a dinner with him, just our subgroup, at some point after we got successful binders, because he was happy. And yeah, and so there's a QR code to our preprint. Yeah, I hope you guys check it out. Yeah, and with that, I'm happy to take any questions.
Thank you. Thank you, Raktim. Thank you very much indeed. Members, if you have a question, you just unmute yourself. I will go first because I have a very question that I
Mm,
think opens up the understanding of this work and what is going on.
mm.
One would naively ask, like, what actually determines whether a protein binds DNA specifically? Like,
Um, well, uh, so the protein binds to DNA specifically. It has to have, you know, a proper charge distribution, for example, because DNA is strongly negatively charged, and that is taken care by MPNN when it's sequence design. And also, it has to have interactions, mainly in the major group of the DNA. If you want to, uh, like for most sequences, where you are chemically recognizing the base by a hydrogen bond pattern that is not amenable to other bases, or like, k-mers. And then there's this other mode of recognition where the shape of the protein or side chains of the protein that recognize the shape of the bound DNA, uh, which is possible for some sequence and not other sequences. Um, but in general, if you want a one-word answer, I would just say it's complex because it's a highly combinatorial problem.
okay, I think when you say that, it's sort of answers with my would be next question, because the paper introduces, like, when the paper starts, it says like sequence specific DNA recognition is
Yeah,
difficult
yeah,
fundamentally.
yeah.
And one would say, okay, what makes it really difficult to be like fundamentally difficult?
It's the fact that DNA is looking like so. So most whenever you are designing protein binders, most of the time, the protein just fitting a hydrophobic region of your target in like a lock and key manner solves your specificity, because not all proteins look the same, right? But all DNA sequence globally looks like a double helix. At least the ones that are in the genome. There's also single-stranded DNA and all of those things, but most of the time, in the genome, you're recognizing a double helix. So what is happening is that the global shape of the DNA is double helix, but depending on every different sequence, it has subtly different chemical signature and subtle different movability. So what we're talking about is 1 angstrom movements and 1 angstrom differentials of arrangements of donors and acceptors. So, so for example, if you're designing an enzyme, you can design a very specific active site. So if this is your reaction, you have your reactant, you have your product, and you can put side chains, which would, you calculate with DFT to generate an active site, and that's exactly how it would look like. But for DNA, because there are so many donors and acceptors, you do not have on your head a solution for what a specific recognition would look like, if that makes sense. So
Yeah.
it is not possible to pre-organize a solution of recognition scheme. You have to let the diffusion model find solutions which are, let's say, basically at a higher dimensional space of intricate arrangement of side chains relative to the DNA, both H1 donor and acceptors, and also the scaffold of the protein relative to small movements of the bound DNA.
Yeah. Um, I've I've not really. interacted with RF diffusion 3, maybe because of computational needs, uh,
Um,
or it says it's, it turns out like in this work, it performs best, like it
yeah.
sort of like gives the best results. Like I've not like an experimental level to
Hmm.
deal with.
Hmm.
Uh, my question is it because like when you look at RF diffusion 2 and 3, 3 has been improved? Is it
Yeah.
because it has learned maybe uh, more about DNA, and that's why it is giving better performance than
Yeah.
others?
So I'll quickly go back to this RFD2 bit. Where is it? So the key difference between RFD 1, 2, and 3 is that um, RFD 1 and 2 are generating the backbone only. So this is not a DNA, but you can see that these are side chains, but everything else that is being generated has only the backbone, right? And that is because it is using this frame representation of the protein, which is different from anything else. Anything else is atomized. While RFD3, like AF3, looks everything as atom. So everything is in the same context. And um, so there is not this mismatch of representation between DNA and protein. And also it is generating the side chains, which is the crucial part. So basically, if you just look at this, wait 1 second, where is it? Um, if you just look at this scaffold, RFD2 scaffold that is being generated, you can actually see right here, it is it is super close to the DNA. So this will clash if there was a side chain there, if that makes sense, right here. And for what happens with RFD3 is that because it is generating the side chains, it has a better sense of for during diffusion sampling, how far different parts of the DNA has to be from the protein backbone. So it has a better spatial packing. So I think this GIF actually shows it very, very nicely. You'd see this atoms being generated. It would come close to the DNA backbone, but then move away slightly. And the DNA also moves based on that. So if you look at, like, for example, right here, so this would be at an ideal distance where it is not clashing. While for RV2, that particular small, like maybe even if it is like, 0.5 angstrom closer to the DNA, the whole thing just doesn't bind anymore, if that makes sense.
Sure,
So,
sure, sure.
so what you said is basically that, but yeah, those are the more specific reasons.
I think my last one is still about specificity. Um, I'm wondering, probably I missed it. How do you optimize this specificity within the entire
Right.
pipeline?
Right. So it comes from two different, so it comes from one is these filters where we make sure that there are so this is, for example, hydrogen bonds to protein side chains that hydrogen bonds to the DNA base, which means when we design say a bidentate interaction with a particular base, and it is also hydrogen bonded with another protein residue, that means we are more certain that that arginine will not rotate away and do something else, if that makes sense. So during diffusion and sequence design and AF3 recycling, some specificity gets conferred through making this kind of things certain. So just have more hydrogen bond network, more supporting interactions, etc. Because if nothing else during the design, it sees the particular sequence you are targeting, and then it is kind of tailor made the side chains of the protein to match that. But that gives you 1 in 1,000 specificity, right? Not 1 in 100. So the 1 in 100 comes from this part here, where you are taking whatever you filtered and then you fold it with off-target DNAs. So you're taking, so this is your intended DNA sequence, and you are taking some of the other DNA sequence and folding the same thing. And then you look at the one that A3 says is the most confident, basically. So, and the confidence generally depends on those kind of things, like, movability, how well it is packed, how many hydrogen bonds networks are there. So this basically gives you an indication of, okay, if it is very confident on its on target compared to the off target, that just means, there is something special it thinks about the on target compared to the off targets. And we just found in the wet lab that that has not the perfect signal, not always whatever AF3 says is happens, but that's why you have to just test 96. Otherwise, you could just test one and get it. Um, but there is enough signal to be able to be the experiment to be done in a plate scale instead of a chip scale, if that makes sense.
Sure. Thank you very much, Raktim. It's good. It's a great discussion.
Thank you.
If there is no questions in the audience, I would like to conclusively, in conclusion, thank Raktim and the members in the audience. Thank you very much, Raktim, and
Thank you.
um, we really can't thank Professor Baker for always, not turning us down, you guys, whenever I reach out. And you produce great work. I know even probably next week there will be a
Yeah.
fabulous paper coming up.
Yeah, I mean, David definitely published a lot of things. This paper has been coming for a long time. I think like the previous paper came out in 2025, but the preprint was in 2023. But I think in general, it took some time for us to actually get DNA binding to a reasonable state. And then, yeah, yeah, um, I'm happy to be here. Thanks for inviting me.
You're welcome. One more question.
Okay.
Somewhere you talked about, not everyone can be the Baker Lab. Wo- If
All
someone
right.
want if someone wanted to do something close to this, even like just replicating it at a smaller scale,
Yeah, you can
following
do it
through the
in a small scale.
code,
Yeah.
what would you say about the computational needs that someone
Yes.
would be put out?
So I believe the previous versions of this work, like the published version with the Rosetta based method and the RD2, which we tried and did not work. Um, those versions were super computationally expensive, but this version is not that much. So if you can run, so it depends on how many targets you have. Generally, I would expect people to use this method to, try to do something specific in terms of function. So let's say you have one target. Um, I think with one target, if you're equipped with, one GPU, you can do this. Um, uh, like in terms of, let's say you have to have um, let's say you want to test 100 things. You can, then you need to maybe have 100 times more of that backbones to start with, because F3 processor is around like 10%. So yeah, I think it is totally doable. It will take you probably a week or something. But this I think the computational bottleneck is not that much, because you can have one GPU, you can let things run for like a month and you'll get things. The real bottleneck is the wet lab bottleneck, and that is lifted here. So if you want to test, you can just do testing in like a plate scale and find something. That's the part where I meant like in bigger labs could do it before, but now I think everyone else can do it too. Yeah.
Thank you. Thank you. Thank you very much, Raktim, and to the team at the Baker Lab and Protein Institute
Mhm.
at the University of Washington. We really can't thank you enough. Thanks for saving up your time and
Okay.
talk to us about
Okay.
this
I'll
work.
stop sharing. Thank you.
All right. So goodbye to everyone and goodbye to Raktim, and
Mm.
see you guys in our next session next week. Bye-bye.